So good morning, everyone. Today I will be giving a presentation for my graduate seminar titled Recurrent Erosion of COA1 or MITRAC15 Demonstrates Gene Dispensability in Oxidative Phosphorylation. Before I start my presentation, I would like to acknowledge Dr. Nagarjun Vijay for his constant support and guidance. I would also like to acknowledge ISER Bhopal for providing the equipments and the funding. I would like to thank all the CG lab members, especially Sagar, Sandhya, Lokdeep and Ashutosh for their hard work and efforts in this project. So with this thanks, I would like to start my presentation. So there is a great diversity of life on Earth, from a single-celled organism to multicellular complex organisms inhabiting almost all the niches which are present on the Earth. So all these organisms need energy for survival, to grow, to reproduce, and even to respond to their environment. They need energy. There is no life as such without energy. So at the cellular level, this energy is provided by respiration. Respiration is of two types, anaerobic and aerobic respiration. So in anaerobic respiration, the process of anaerobic respiration is called glycolysis. In this, the one molecule of glucose is degraded into pyruvate and a lot of ATPs are formed during this process, along with the molecules of NADH. In aerobic respiration, which takes place inside the mitochondria, a lot of NADH, are formed by the process of citric acid cycle, and these NADH and FADH2 pass on to the oxidative phosphorylation, which happens inside the inner mitochondrial membrane, and these molecules are then converted into ATPs by the process of oxidative phosphorylation. So what happens exactly in oxidative phosphorylation or OXPHOS? So in this, a chain of mitochondrial inner membrane embedded proteins or complexes, which are four complexes, pass electrons from one complex to another and through some redox reactions and form a proton gradient. This proton gradient is then released. This proton gradient is then released by the fifth complex, which helps in the formation of ADP to ATP. So these four complexes are extremely important in the OXPHOS pathway. The energy that is derived from OXPHOS as well as glycolysis is required by the skeletal muscles for locomotor abilities. So based on their requirement of energy, they are divided into two parts, type 1 and type 2. So type 1 fibers are OXPHOS dependent fibers, while type 2 fibers are glycolytic fibers. Glycolytic fibers are those that depend on the anaerobic respiration. These are the 2B and the 2X type, while OXPHOS dependent fibers are type 1 and type 2A. OXPHOS dependent fibers are also known as the slow twitch fibers. They are aerobic in nature, but they do provide energy, but they fatigue very slowly. As compared to the slow twitch fibers, the glycolytic fibers, also known as the fast twitch fibers, are dependent on the anaerobic respiration. They fatigue very rapidly, but they provide a burst of energy at one go. So as you can see in this image, the sprinters, they require a burst of energy for a short duration of time. For them, they rely mostly on the fast twitch fibers, while the marathoners, which require a constant stream of energy and require the resistance to fatigue, they depend on slow twitch fibers. Similarly, in nature, species which are extremely energy intensive, which have extremely energy intensive adaptations, which require a lot of jumping and sprinting, they require mostly fast twitch fibers. While the species which do not require, which need a constant supply of energy, they are more dependent on the slow twitch fibers. So, as for example, we know that the fastest mammal on Earth is cheetah. It can attain a high speed, but for a very short duration of time. It can accelerate rapidly, but gets tired very quickly. Most of the apex predators, which are felines, felids, they have a lot of glycolytic fibers in their skeletal muscles, And so they can run or accelerate extremely fast, but they get tired very quickly. As compared to the felids, the other predators like the canines, the wolves, they hunt in packs. They can run at a large distance, but they cannot accelerate that fast. This is the talk about, so we have just talked about predators. What happens in prey species? Now, prey species need to escape the predators. So they, along with a high energy, they require resistance to fatigue. So it was found that in their skeletal muscle composition, both OXPHOS and glycolytic fibers are more. In the case of felines, there are more glycolytic fibers, while in the case of cervids, that is the deer family, has both OXPHOS and glycolytic fibers. So like in the case of rodents, they have more 2B fibers than larger mammals. In fact, 2B fibers are absent in human. So they have more glycolytic fibers, which give them a short burst of energy, and they can run very fast, but for a very short duration of time. As most of us know the story of the hare and the tortoise. So the hare and the tortoise, it's a children's tale in which the hare and the tortoise decided to compete, but after running for a short while, the hare got tired and slept in the middle, and that's why the tortoise won the race. So this happened probably, though this is a children's tale, but this probably happened because the rabbit has a lot of glycolytic fibers instead of OXPHOS fibers. So in the case of birds, the ability to fly is a distinctive feature in birds. So the large amount of energy which is required for this flight is necessitated by a high metabolic rate in birds. So this high metabolic requirement has been met through various adaptations in flight. So common swifts are long migrants, and they can stay in flight without any stop for 10 months non-stop. And for this, they require a lot of energy for a long duration of time, and they require the resistance to fatigue. And it has been found that they have more more oxidative fibers, while in the case of passerines, small passerines, they need to hover at one place. They require a lot of energy to hover at one place to derive the nectar from the flowers, and that is why it has been found that they also have a lot of OXPHOS fibers. In contrast to this, if you look at the Galliformes, so this is a group of Galliformes, also known as the land fowls. Chicken is one of those land fowls. We have peacocks and turkey also in this group. These group of birds do not go for long flights. They require a short burst of activity for a short flight. And in a study, it was found that Galliformes contain mostly glycolytic fibers. So what happens in the OXPHOS pathway? OXPHOS pathway it's under most of the proteins in OXPHOS pathway are under strong purifying selection because it is very essential for survival. But some genes get quickly positively selected in the adaptations which are extremely high energy demanding. Like in the example of polar bear, it requires a lot of energy to survive in the cold polar Arctic environment. So the genes in the OXPHOS pathway are positively selected in this case. While in the case of dogs, which got domesticated, now the reliance on energy has been reduced. So it was found that there is a relaxed selection on the OXPHOS proteins. So it's a common fact that as a consequence of the relaxed selection, gene loss can happen. So a gene can get lost when it is not in use because it is under relaxed selection. What is exactly COA1 or MITRAC15 and what is its role in OXPHOS? So COA1 is also known as the cytochrome C oxidase assembly factor 1. It is a component of the MITRAC complex, which is found in the inner mitochondrial membrane. So MITRAC is also known as the mitochondrial translation regulation assembly intermediate of cytochrome C oxidase complex. Cytochrome C oxidase is the fourth complex that I just showed earlier in the OXPHOS pathway. So MITRAC is basically a translational regulator of the which helps in the biogenesis of complex 4. It regulates the translation of mitochondrial encoded components, as well as it helps in the assembly of the nuclear encoded components which are imported inside the mitochondria. So it was found that this gene COA1 or MITRAC15 has a very mild expression, and overexpression of other genes can compensate for its mild effects. In several gene knockout studies, it was found. But very interestingly, in one of the studies by Vanderlee, it was found that it is positively selected in primates. So this proves that even though it has a very mild effect, it does confer some kind of fitness to the organisms, and that is why it is getting positively selected in the primates. So this knowledge leads us to postulate that what is happening to COA1 in these 3 groups of organisms where there is more reliance on the glycolytic fibers instead of the OXPHOS fibers. So we came up with a hypothesis that there is relaxed selection on these 3 groups, which results in the loss of COA1 in felids, in rodents, and galliforms based on the increased glycolytic fibers. To search for this, we used various types of tools. First, we validated the COA1 annotation. So there are more than 300 vertebrate genomes available on Ensemble and NCBI showing the annotation of COA1. This annotation was validated using gene synteny and sequence identity. So gene synteny is basically the gene order on a particular chromosome and shows the evolutionary history of the gene. So by looking at the gene synteny, we can ensure that there are no paralogs and the sequence which we are studying is correct. We also used MSA and the RNA-seq data to check for the sequence identity so that we can be sure that this is the sequence which is correct. It can be ensured that the sequence under study have not diverged a lot with the help of both MSA and RNA-seq data. After the verification of sequence from Ensemble and NCBI, we tried to verify the COA1 gene by using in the raw read data. If there is any ORF disrupting changes, we checked for any ORF disrupting changes in the raw reads using BLASTM. After this, we see if there is what is the transcriptional status, if there is any transcription happening in that gene using the tools like Star Mapper, and we visualized the results using IGV. We did further molecular evolutionary analysis. To do the phylogenetic analysis, we downloaded a time-calibrated tree from PineTree. We also tested for relaxation using 2 programs like Relax from HiFi and CodeML from PAML. After this, if there is a loss in gene, we try to estimate the gene loss timing using the method used by Meredith et al. In 2009. We also estimated the GBGC using MAP, NH, and FAST. And in the cases where we try to see if there is any alternative splicing happening, we try to see the RNA binding site. We try to predict the RNA binding sites using RBP map. So if there are any homologs present, to look for any homologs so that the function of COA1 can get compensated, we try to find the homologs, and for this we use several tools which I shall be discussing in the results. So in results, we found gene loss in Galliformes. So first of all, we confirmed the synteny of COA1 in Galliformes, and it was found that the synteny is conserved. Synteny is basically the gene order. So it was found that the synteny is conserved in Galliformes as well as the Enseriformes. Here, Enseriformes are used as an outgroup for the comparisons between these 2 groups. And when we checked the transcriptional status of this Enseriform, we found that in most of the genes, wherever the data was available, the red box are showing the transcriptional status, And we found that they are transcribing robustly, the Enseriformes. But in the same case, when we checked the transcriptional status in the Galliformes, we found that in wherever the data was applicable, this gene is not transcribing except for electoral athamy, which is showing some transcription in the blood tissues. So on further analysis, when we checked their genes, we saw that there are several substitutions showed here by yellow lines, and there are certain stop codons also shown here by the whites, and insertions and substitutions. So there were several gene disrupting mutations throughout the Galliform, and a lot of independent losses of COA1 was found throughout the Galliform group. So we confirmed this using the PacBio sequence of Gallus gallus. We checked in more than 22 tissues of Gallus gallus the transcriptional status of this gene, and it was found that it is not transcribing. So we can very confidently say that this gene is lost in most of the Galliforms. And we used chicken because chicken is one of the most well annotated genome. So in the PacBio data also, we confirmed that this loss is there. So after confirming in Galliforms, we moved ahead to rodents. In rodents, we found that there were several gene disrupting mutations in 4 species of Sciuridae, which were shared. And so this means that this loss happened before the divergence of these 4 species. We also found independent losses in the beaver and in rabbit, also in the naked mole rat, which is Heterocephalus glaber. In rabbit, it was seen, we checked its transcriptional data, and we found that this gene is not transcribing. And so after this, then we went ahead and checked with Castor canadensis, which is a North American beaver, and we found several independent mutations like deletions in the second exon or premature stop codon, and substitutions we found in beaver. After this, we also checked in Heterocephalus glaber, that is in the naked mole rat, if there are some certain gene disrupting mutations. So here we found that it doesn't have a start codon, and also there is a gene disrupting mutation, which is a deletion of a single base pair in the first exon. Even after this, we found that there is some transcriptional activity going on in naked mole rat. So we postulate that probably this is a long non-coding RNA, which is not coding for any protein, but further analysis needs to be done to establish this fact. And we try to further look into naked mole rat. Then we found that there is a duplication in naked mole rat, which I will be discussing later. So when we along with these gene disrupting mutations, when we checked for the synteny, we found that the synteny is not conserved. And rodents are very famous for the presence of a lot of EBRs, which are evolutionary breakpoint regions. So what are exactly evolutionary breakpoint regions? So this is not a physical entity. It is basically an analytical construct which arises when we are comparing 2 genomes. So when we are comparing 2 genomes based on the methods and assumptions, this is a construct that we analyze in our study. So there is something called a homologous synteny block, which is a conserved gene order. So this is a contiguous region of 2 homologous synteny blocks here, and this is a conserved gene order which are found in other genomes as well. So when we are considering homologous synteny blocks as a unit, we find that there are breakpoints in these homologous synteny blocks. So what are breakpoints? So 2 contiguous chromosomal segments in one genome, which are orthologous to different segments in another genome, but are not contiguous to each other. When a continuous region is broken up in the second genome into different parts, then we say that there are breakpoints present. But this breakpoint is not a single point, that there are 2 base pairs and at that exact point there is a break happening. It doesn't happen this way. This is actually a lengthy region where this break happens. And this region, because it's a lengthy region, it's known as a breakpoint region. So evolutionary breakpoint regions are the regions where the synteny has been disrupted by chromosomal reorganizations. This region basically does not align well to either of the two segments of the second genome or their flanking sequences. So in one of the studies by Fitzgerald and Bateman, they found that EBRs are, rodents are enriched with a lot of EBRs. So Fitzgerald and Bateman found evolutionary breakpoint associated gene deletion. So they found that because of this chromosomal rearrangement in this region, evolutionary breakpoint region, it has led to gene deletions of several genes in rodents. And they found that along similarly to these genes, the STK17 is also lost because it is present on an EBR. In the conserved gene order in all the vertebrates, we find that STK17 is right upstream of the COA1. So we think that this region is present in COA1 is present in EBR. So on further analysis, we found that the synteny is not conserved and there is a loss of COA1 in a lot of species, and there has been a movement of this conserved gene order in a lot of species. So for the sake of this study, I will be using a very non-technical term for the sake of convenience. I will be calling these species as the EBR species, which have lost COA1 because of the chromosomal rearrangement, as this COA1 is present in EBR region. And I will be calling these species as the pre-EBR species, which have not lost COA1 or has kind of a conserved sequence because there has not been any pre-chromosomal rearrangement. So these are the pre-EBR species and these are the EBR species who have lost COA1. So in the zoomed in view, if I show you the pre-EBR species, we can see that this region is conserved. The left flank of these three genes, which is STK17A, COA1 and BLVRA, the left flank of these genes are conserved, while the right flank are in different orders like O1, O2, O3, O4, and O5. So there are 4 different orders on the right flank. But in the post-EBR species or the EBR species, if you can see that this COA1 has moved to a different region between PTPRF and HY1 gene, while BLBRA has moved to a completely different location. And these sequence of genes like HECW1 and MRPL32 have moved to a different gene order like O9. And these genes have moved to a different gene order like O6. We only found remnants of COA1 in between these two genes, which were exon 1 and intron 2. Only remnants and parts of this COA1 gene was found in between these two genes. So the question arises that how are we sure that this is not a paralog and this thing is not present in the pre-EBR species as well, this remnant of COA1. So for this, we checked if there are some remnants of COA1 present in pre-EBR species as well, and we found that there is no such remnant present in between these two genes in pre-EBR species. So we can very confidently say that because of there is a movement of BLVRA and HCW1 also. So we can very confidently say that this is the original COA1 which has been lost as it was present in the EBR region or the evolutionary breakpoint region, and it has been lost in all these species. So when we tried to check the naked mole rat, the status of the naked mole rat, we found that in the raw read data, there were 2 distinct haplotypes present in exon 2, and there were no second copy present on exon 3, exon 4, and of exon 1. So we are postulating that probably this is because this gene has diverged a lot, or maybe there is some sequencing problem, and we need to analyze this more. While studying the raw read data, when we visualized it in IGV, we found that several raw reads were aligning, distinct haplotypes were aligning in the case of human as well as tiger. Human, which is primates, we looked in the case of human, we found that both in the case of human and tiger, the pseudogenic copy is the one copy is pseudogenized and the other copy is transcribing. So the pseudogenic copy has moved to a different location while the transcribing copy is present in the syntenic location, and it is perfectly transcribing. On further analysis in primates, we found that there is an extra N-terminal extension in the old world monkeys. So there is an N-terminal extension, but in the case of Cercopithecidae, we found that there is no original start codon. This original start codon has been lost, while in Catarrhini, there is the original start codon is still intact. No such thing has been found in their closely related New World monkeys. If you can see here, along with the primates, there is certain intron-exon rearrangement happening in Feliformia as well. There is a lack of exon 3 in Feliformia. So on further analysis, we found that in this particular clade, except for suricata, there is a loss of single base pair in the second exon. So this loss in all of these felids has led to a change in the end phase of the exon 2. And if there is an inclusion of the conserved exon 3, which I'll be calling as a dog-like exon 3. So this is a conserved exon 3 which is present in all the carnivora. This conserved exon 3, if its inclusion because of the phase change at the end of the exon 2 is resulting in the premature stop codons and disrupting the ORF. So we found that this felines are skipping this third exon and in alternatively splicing it and directly jumping to the fourth exon. In fourth exon, however, there is all of them share a loss of 2 base pairs, and because of this, the stop codon is changed at the end of it, and there is an extension of the fourth or the third exon in the case of cats. So this fourth exon or the third exon of cats are extended, and that's why the ORF, the length of the ORF is comparable between Canis lupus and all the other all the felines that we're studying. So, but in the case of when we try to study their transcriptional activity, all of them were transcribing, but this Puma concolor and Felis catus, which is the cat, and Puma, they showed proto-cat-like exon 3's transcription in this region. However, most of the transcribed regions were skipping this exon, and they were directly taking directly going to the exon 3. So in this case, when we looked at the transcriptional activity, we also found that there is no transcriptional activity happening in cheetah. On further analysis, we found that there is a substitution happening from C to T in this case, and that is why this gene has been completely lost along with single base pair deletion in the exon 2. This gene has been completely lost in cheetah. So we can very safely say that this gene has been now, we can say that this gene has been lost in the cats, the cheetah, the Galliformes, as well as the rodents. So after this, we try to look for we try to find the homologs which can compensate its function. So homologs were identified mainly by the similarities which are observed in the sequence, their structure and their functions between the proteins. So firstly, to find homologs based on the sequence, there are several challenges to find homologs. So For example, many proteins that are functionally and evolutionary related, but their sequences have diverged so much that they're not easily identified by direct sequence comparison. So hence highly sensitive tools like SciBlast and HSBlitz were used to search for the proteins iteratively in the database to find distant homologs using human COA1 amino acid sequence as a query. So the top search results of this homologs which were identified uh, were visualized and clustered using clans. So as you can see that in this cluster, the top hits are dominated by TIM21 here encircled in the red, and COA1 from different organisms such as bacteria, fungi, and plants. So these two are plants. So this suggests that TIM21 is a homolog of COA1. To further verify whether the database matches are homologous, we also evaluated the structure and the function of TIM21. So to obtain the secondary structure predictions for COA1 and TIM21, we used HeliQuest 2 and Proter Web Server. On this side, on the left-hand side, we can see the predicted secondary structures of COA1 and TIM21. So it was observed that COA1 and TIM21 has comparable secondary structures. So, for example, they both have intra-membrane region consisting of helix, which is this marked in green. In this figure on the right-hand side, we can see that COA1 and TIM21 protein sequences are aligned along with their secondary structures as query and as template, respectively. So the first line represents the secondary structure such as H for helix, E for coils, and E for beta strand, and C for coils. In the second line, amino acids are colored based on their physicochemical properties such as charge. So now if you look at the secondary structure part of the alignment, it shows regions with the most substantial homology in both the proteins. Like this region in the box highlights the helix region in both the sequences. In addition to the sequence and structure homology, both COA1 and TIM21 are known to play prominent roles in mitochondria. TIM21, TIM21 is a component of TIM23, which is the translocase of inner mitochondrial membrane. TIM21 helps in the translocation of the nuclear encoded protein components across the mitochondrial inner membrane towards MITRAC, which ultimately assembles these proteins and the proteins from the mitochondrial encoded proteins and helps in the biogenesis of complex 4. TIM21 is also a part of the MITRAC. So we can very clearly see that they both play a very essential role, but they are very distinct roles in the inner mitochondrial membrane. So we can safely assume that TIM21 is a distinct homolog of COA1. There is a lot of new members in TIM and the TOM proteins. TOMs is the translocase of the outer membrane. So these two complexes have been enriched by a lot of gene duplications, creating a rich library of inner mitochondrial proteins which are involved in a mitochondrial import system. So hence it is very possible that COA1 has resulted from the duplication of TIM21 and then it has diverged a lot, having a distinct function different from TIM21. So finally, we can say that COA1 and TIM21 are distinct homologs, and TIM21 has been duplicated, which got diverged and has formed COA1. Probably this duplication of TIM21 might have occurred during the movement of TIM21 homolog from the mitochondria to the nucleus. We also established independent COA1 gene losses in cheetah, galliforms, and rodents. In the case of felids, we saw that there was an alternative splicing of the third exon in response to the gene disruptive changes in the second exon. But what is happening at the protein level, it needs to be confirmed because we expect that the protein might have changed a little. So its function and its its function needs to be checked at the proteome level. Very interestingly, we found that birds which are flightless or have limited ability to fly has an intact transcribed gene. So, therefore, the loss of the COA1 gene appears to be associated with changes in the skeletal muscle fiber composition rather than the vice versa. So, if we identify what are the what are the changes in the genes, or what are the genetic changes which are determining the changes in the muscle fiber composition, and what are the sequence of events, we might be able to understand more the phylogeny and the phylogenetic history of what is happening at in COA1, why it got lost in those organisms. COA1 is also an oncogene which is involved in colorectal cancer. So the loss of this gene in several organisms, so this could actually reduce the risk of cancer. So further identification of gene loss events in birds and in other organisms might reveal other oncogenes which are lost, and this can pave a new way in various cancer studies. In a study by Garagnani et al., very recently, it was found that it was a study between supercentenarians and so people who are above the age of 100 and people who are below the age of 100. They took two groups, and it was found that in the Jiva study, it was found that the people who were supercentenarians showed low transcription of COA1, low expression of COA1 gene. So this means that COA1 gene, or genes which are involved in the mitochondrial OXPHOS pathway are involved in providing health and longevity. So we also found, in our study found that COA1 is lost in beaver and naked mole rat, which these two species are model organisms to study longevity. So further studies of COA1 gene loss in beaver and naked mole rats will illuminate the relationship between the longevity and the loss of COA1, or how COA1 is involved in the loss of in providing longevity. This study also illuminates the role of EBRs, that is the evolutionary breakpoint regions, and the gene loss while constructing the evolutionary history of rodents. We obviously need more data, and high quality near complete vertebrate genomes will further aid in the large scale identification of gene loss events across the vertebrate phylogeny. Thank you.